Hi, and welcome to Booga Bio. So, today we'll learn about cell signaling, and the first component of any cell signaling is a ligand or a signal molecule, which can be a photon of light, or a taste or smell molecule, or a hormone. And as the name suggests, it's a signal molecule, which means it acts as a signal or a key to start any signaling process. And this is our receptor onto which the signal molecule will join. And specifically, this is a GPCR receptor or G protein coupled receptor, from which you can understand that this receptor is coupled with G proteins. And GPCR is a 7-pass transmembrane protein, which means it passes onto the lipid bilayer 7 times. And this right here is our G protein, which is made out of 3 subunits, alpha, beta, and gamma. And onto the alpha subunit, there's a GDP molecule which is attached onto it. And if you look more closely, you can see that it's only the alpha and gamma subunits which are attached onto the inner leaflet of the lipid bilayer, and the beta subunit is just hanging onto the gamma subunit. So what this activated GPCR receptor does is that it replaces the GDP with GTP, which activates the G protein as a whole, so it can initiate further downstream processes. To get a deeper understanding, we can say that the alpha subunit is in an off state when it is bound to GDP, and when the receptor or GPCR receptor gets activated, it acts as a GEF or guanine exchange factor, which replaces the GDP with GTP, which turn ons the alpha subunit. And to balance out things, that the system doesn't crashes out, the alpha subunit itself also act as a GAP or GTPase activating protein, which removes the phosphorus from GTP, which converts again GTP back into GDP.